Okay, good morning. I'm discussing microscopic techniques. There are three kinds of microscopic techniques: optical microscopic techniques, electron microscopic technique, and scanning probe microscopic technique. Uh, we discuss here electron microscopic techniques. Electron microscopic techniques and electron microscope is a type of microscope where light source is an electron beam. Light source, electron beam that has been accelerated. This unique kinds of microscope creates the image with a high resolution, can magnify objects as small as nanometers. The range of capture on a phosphorescent screen with the control application of the electron in vacuum. Working principle of electron microscope. Electron microscope use signals arising from the interaction of an electron beam with the sample to obtain information about the structure, morphology, composition. Electron microscope use signals arising from the interaction with the electron beam with the sample to obtain information about structure, morphology, and composition. The electrons gun generated electrons. The two sets of condenser lens focus the electrons beam on the specimen and then into thin tight beam to move electrons down the column and accelerating voltage 100 kilovolt to 1000 kilovolt is applied between the tungsten filament and anode. The specimen to be examined is made extremely thin, at least 200 times thinner than those used in the optical microscope. Ultra thin sections of 20 to 100 nanometer are cut, which is already placed on the specimen holder. The electronic beam passes through the specimen and electrons are scattered depending upon the thickness of refractive index parts of the specimen. The denser region in the specimen scatter more electrons and therefore appear darker in the image since fewer electrons strike that area of the screen. In contrast, the transparent regions are brighter. The electron beam coming out of the specimen passes to the objective lens, which has high power and forms the intermediate magnified image. The ocular lens then produces the final further magnified image. The specimen to be examined in the made extremely thin, less 200 times thinner dose in optical microscope. Ultra section 20 to 100 nanometer are specimen holder. The electrons beam passes through the specimen and electrons scattered depending upon the thickness or refractive index of different parts of space. The denser region in the specimen scatter more electrons and therefore appear darker in the image. Since fewer electrons strike at that area of the screen, the electrons being coming out from the specimen possess the objective lens, which has high power and forms the intermediate magnified image. The ocular lens then produces the final further magnified image. See, this is the instrumentation of electron microscope of the scanning electron microscope. Same, and this is the electron gun. This is the condenser lens. This is the objective lens. This sample, and this scanning foils. This anode. Okay, I am discussing about as a whole the working principle of electron microscope. Thanks for watching and request you all like, comment, share, and subscribe. Thank you for your patience. That is now. Bye bye.